Good afternoon, dear learners. I am Deepali Kulkarni, Assistant Professor, Yash Institute of Pharmacy, Aurangabad. Today, topic of our webinar is beta oxidation of fatty acid. After watching this webinar, learners will be able to remember different types of oxidation reactions, understand process of beta oxidation of fatty acid along with energetics of beta oxidation. As all of you know that lipids are non-polar organic compounds which acts as regulatory substances. Lipid catabolism, that is metabolism of lipid, or in the simple words we can say that breakdown of complex organic compound into simpler one. This is highly energy releasing process, and energy produced through this process is known as lipogenesis. Beta oxidation of fatty acid is breakdown of fatty acid to acetyl coenzyme A. End product of each cycle is the fatty acid, which is shortened by 2 carbons and acetyl coenzyme A. Reaction site is beta carbon, that is third carbon from the thioester carbon. It happens in the mitochondrial membrane. Acetyl coenzyme A is directly fed into the Krebs cycle for the further oxidation reaction, where it gets converted into carbon dioxide and water molecule with the generation of energy. Oxidation of fatty acid usually occurs in various tissues and organs including liver, kidney and heart, but it does not occurs in the brain. Following are some types of fatty acid oxidation: alpha oxidation of fatty acid, beta oxidation of fatty acid and omega oxidation of fatty acid. The first step of beta oxidation includes activation and transportation of activated fatty acid to mitochondria. Fatty acid is combined with coenzyme A in presence of ATP, where magnesium acts as activator. There is formation of fatty acyl coenzyme A, AMP and 2 moles of inorganic phosphate. This reaction is coupled to cleavage of ATP. Acyl coenzyme A synthetase, which is a member of isoenzyme family, acts as catalyst for this reaction. Synthesized fatty acyl coenzyme A is transported with carnitine to inner mitochondrial membrane. Now next step is transport of fatty acyl coenzyme A from cytosol into mitochondria. The activation of fatty acid takes place in the cytosol, and actual site of beta oxidation is mitochondrial matrix. So once there is formation of fatty acyl coenzyme A, this long chain acyl coenzyme A traverses in the inner mitochondrial membrane. A special transport mechanism is used for this purpose, which is known as carnitine shuttle. The detail of carnitine shuttle is represented in the diagram along with structure of carnitine. This is actual process of carnitine shuttle. This is a rate limiting step where acyl group transferred to carnitine and there is formation of acyl carnitine. Carnitine transferase 1. This is the enzyme which is present in cytosol carried out this transfer in the outer mitochondrial membrane. Next step is acyl carnitine is then shuttled across the inner mitochondrial membrane in presence of enzyme translocase. During this transfer, the acyl carnitine is travelled through intermembrane space which is represented in the diagram. Now acyl group is transferred back. Coenzyme A matrix by carnitine acyl transferase 2 enzyme which is present in mitochondrial matrix, and carnitine returned to cytosolic side by enzyme translocase. This is overall reaction flow chart which occurs in inner mitochondrial matrix. We have taken palmitic acid as an example. There are major 4 steps which are also known as proper beta oxidation steps, where activated fatty acid or fatty acid acyl coenzyme A undergo dehydrogenation reaction in presence of enzyme acyl coenzyme A dehydrogenase. Here FAD acts as hydrogen acceptor and converts into FADH2. The structural changes which occurs in palmitic acid is highlighted by pink color in this flow chart. After dehydrogenation, there is formation of 1 pi bond with the cleavage of 2 sigma bond, and the name of newly formed compound is trans delta 2 enoyl coenzyme A. This trans delta 2 enoyl coenzyme A undergoes hydration reaction with the addition of water molecule in presence of enzyme enoyl coenzyme A hydratase, and there is formation of L beta hydroxy acyl coenzyme A. Now this L beta hydroxy acyl coenzyme A undergoes dehydrogenation reaction. In this reaction, NAD plus acts as hydrogen acceptor and there is formation of beta ketoacyl coenzyme A. Beta ketoacyl coenzyme A finally cleavaged in presence of enzyme acyl coenzyme A acetyl transferase. This is member of thiolase family, and there is formation of acetyl coenzyme A and another fatty acyl coenzyme A, which is 2 carbon shorter than that of palmitic acid. The name of compound is myristoyl coenzyme A. In figure B, there are shifting of C16 carbon chain in first step to C14, and as later on with the continuation of 7 cycles there is formation of acetyl coenzyme A 2 molecules. Now proper beta oxidation which is carried out in mitochondrial matrix, there are 4 steps of beta oxidation which occurs in mitochondrial matrix. Step 1 is oxidation by FAD linked dehydrogenase. Step 2 is hydration of compound in presence of enzyme hydratase. Step 3 is oxidation by NAD linked dehydrogenase enzyme, and step 4 is thiolytic cleavage in presence of enzyme thiolase. So first reaction is oxidation of acyl coenzyme A. The reaction is catalyzed by enzyme acyl coenzyme A dehydrogenase. It gives alpha beta unsaturated acyl coenzyme A, which is also known as trans delta 2 enoyl coenzyme A. FAD acts as hydrogen acceptor. The structural changes are highlighted with the formation of box. As the name alpha beta unsaturated compound is given because there is presence of alternate double bond in the structure. Second reaction is hydration reaction. Newly formed double bond is hydrated and there is formation of L beta hydroxyacyl coenzyme A. This reaction is catalyzed by enzyme enoyl coenzyme A hydratase. Third reaction is oxidation reaction where beta hydroxyacyl coenzyme A oxidized to produce beta ketoacyl coenzyme A. This is NAD dependent reaction, and this reaction is catalyzed by beta hydroxyacyl coenzyme A dehydrogenase enzyme. There is formation of another thioester carbonyl carbon in this structure. And last step, that is fourth reaction, is cleavage. 2 carbon fragment is detached from the compound by splitting the bond between alpha and beta carbon. Thiolase enzyme acts as catalyst, and there is synthesis of another activated fatty acid which is 2 carbon shorter than that of parent fatty acid. Now energetics of beta oxidation. Beta oxidation of palmitic acid will be repeated 7 cycles. That is, there is formation of total 8 molecules of acetyl coenzyme A. In each cycle, FADH2 and NADH plus H plus is produced and they are transported directly to the respiratory chain. One molecule of FADH2 is equivalent with 2 ATPs, whereas one molecule of NADH plus H plus is equivalent to 3 ATPs. So in 7 cycles there is formation of 5 multiplied by 7, that is 35 ATPs. Each acetyl coenzyme A, which is further oxidized in citric acid cycle, or Krebs cycle gives 12 ATPs. So 8 molecules of acetyl coenzyme A individually gives 12 ATPs, combinedly become 96 ATPs. 2 ATPs are utilized during the activation of fatty acid. Hence, the net gain of beta oxidation is 35 ATPs plus 96 ATPs minus 2 ATPs, which is equals to 129 ATPs. That is, huge amount of energy is generated with the entire beta oxidation of one molecule of palmitic acid. So, summary of our today's webinar is the release of acetyl coenzyme A leaves an acyl coenzyme A molecule which is shortened by 2 carbon. Acyl coenzyme A molecule is now substrate for oxidation starting with acyl coenzyme A dehydrogenase. Repetition continues until all the carbon of original fatty acid is converted into acetyl coenzyme A. In the last round, a 4 carbon acyl coenzyme A, that is butyryl coenzyme A, is cleaved to 2 molecules of acetyl coenzyme A, and total energy gain after completion of one cycle of beta oxidation is 129 ATPs. So, thank you dear learners. If you are having any query, please feel free to ask.